folks, but Yazzie, do you want to start us off?
Yes. Hello, everyone. Thanks so much for joining. This is the Enlightened Soil webinar, and tonight we have a special guest, Dr. Jerry Hatfield, and we're really excited for you to join us as we have this conversation with him and move into a Q&A later in the evening. So just to start off with an agenda of how the hour will go, I'm going to start by telling everyone the origin of Enlightened Soil Corp, which is the company we work for. And then Camille will go over how EnSoil Algae works. And then we'll start our conversation with Dr. Jerry Hatfield. He'll be talking about how to improve production efficiency. And around 7:30, we'll start our Q&A, but throughout the session, just enter your questions in the chat and then we'll get to them at that point. And finally, it's our hope that whatever you learn from here, you'll be able to spread to your communities and help get this regeneration thing going. Okay, so I'll start with the story of Enlightened Soil Corp. So it starts with this man on the left here, Dr. George Taylor. He's our founder, and he is a former cardiologist at MUSC in Charleston, South Carolina. And once he retired, he decided to combine his two lifelong passions, gardening and healing people. So we purchased some land on Johns Island, South Carolina, which is where we're based out of, and established a nonprofit called Sweetgrass Garden. And the purpose was to grow chemical-free, nutrient-dense food for local communities in need. When he got there, though, realized the land needed some help and began deep diving into chemical-free soil amendments he could use that were also inexpensive, being it's a nonprofit. And that's when he stumbled across our best friend, Chlorella vulgaris. So Chlorella vulgaris, there's been research since the 70s of it being used, but it's presumed that it's been used across the world for centuries as a replacement for fertilizer. So Dr. George Taylor began using Chlorella vulgaris on sweetgrass, and the results were profound in soil health and crop production. He was pretty amazed and asked the question that started this all, which is, why is this not commercially available? And discovered the answer to that was threefold. First, fertilizer was still somewhat affordable at the time because this was in 2018. Two, algae didn't seem to really be in the first wave of biologicals that was coming out. And three, the most important one, no one had yet just figured out how to bottle these photosynthetic cells and keep them living. And that is what Dr. George Taylor and a brilliant team sought out to do. From 2018 to 2020, we're just working on this grow process, simultaneously working with local growers and sharing the algae with them. Actually, some people on our team were some of those people that were trying out the algae and saw the benefits. And then finally, October 2020, established the grow and bottling process to keep these beautiful living cells alive. And that's when Enlightened Soil Corp was formed. So we're still relatively young. We just came up on 3 years. So just sharing this trial, it's a very important one, also our first one. It was done at Iron Horse Farm in Johns Island, and the question was, does reduction of nitrate inputs in pasture land treated with Chlorella vulgaris result in cost savings and healthier soil and grass? And you can see from the pictures, it was a huge success. The number one ranked pasture was treated with the algae alone because they did combinations of fertilizer plus algae, just fertilizer, algae. The number two ranked pasture was treated with algae plus 25% of the usual NPK input. And again, this trial is really important. It really speaks to the heart of our mission, which is to help the world transition away from chemically dependent agriculture And it's an interesting note to point out the pond near near the farm had really bad algal blooms. And now there's none. So yeah, that was really special, and we've only done more incredible things since then. And here's a team, minus a few. So passing over to you, Camille.
Good evening.
Oh, I muted you by accident. I meant to mute me.
Hi, everyone. Thank you so much for being here. We really appreciate the attendance and the interest in what we're doing. So I'm going to explain a little bit about the mechanisms of the algae before we turn it over to Dr. Jerry Hatfield. So as Gazi said, the strain of algae we are working with is called Chlorella vulgaris. This is a micro green freshwater algae that naturally occurs in all ecosystems around the planet, and it is significantly older than we humans are. So this strain of algae works as a powerful biostimulant, and it encourages the natural processes of nutrient cycling and soil microbial activity to help stimulate the plant growth. There are four main sort of mechanisms or ways that we have noticed through our data and research that the algae is working. So I'm going to list those here, and then I'm going to explain them with an infographic on the next slide. So first, the algae stimulates and grows the microbial population and diversity. Second, it increases plant nutrient uptake and stimulates the overall plant growth. Third, it enhances chlorophyll, which is that green pigment that you see in a green plant, and improves the photosynthetic capacity, so the ability for it to grow and exchange gas, draw down carbon, and use sunlight to provide itself energy. And then lastly, it increases plant tolerance to stress, specifically abiotic stress, such as drought or high salinity, and it increases the soil water retention capacity. So we have some really great data in regions with high drought. So now I'm going to explain a little bit more in depth using a comparison with synthetic NPK fertilizer. So for those of you that maybe are not growers or don't know what this means, your traditional fertilizer is synthetically produced, and oftentimes the nutrients that are being added are nitrogen, phosphorus, and potassium. So that's what the NPK stands for. So to follow along with me, we'll start on the bottom right-hand corner. And with EnSoil, you apply it. It is a spray. You can apply it in drip irrigation or a watering can, flood irrigation, any way you can get water to your soil and your plants. So when it is applied, the application stimulates the growth of the microbial population and the diversity. So we see more species, and we also see greater populations, more numbers. Second, the bacteria, so some of these soil bacteria, these microbes, are helping to fix or convert the atmospheric nitrogen into a usable form for the plant, and the plant is able to take up those nutrients through the roots. And then third, when the EnSoil is applied, some of it's going to land on the leaves. And what happens when it lands on the leaves is that the chlorophyll, that green pigment, is enhanced in the plant, and it helps photosynthesize that plant more efficiently. So what we see as a result of that is that there's more carbon that's drawn down and released into the soil. This typically comes back in our data as an increase in soil organic matter or water extractable organic carbon, which is a fancy way of saying the carbon that is available for the microbes. And then lastly, the EnSoil increases the plant tolerance to stress, again, such as drought or high salinity, and the water retention capacity. So we see an increase and have that plant be able to use that water. So the difference, to have a comparison here with synthetic fertilizer, is that when we apply fertilizer, we are providing an external source of nutrients. So these are synthetically, chemically produced. And for this example, I'm going to use nitrogen. So the form of nitrogen is typically inaccessible to the plants without this community of microbes to fix it. And fixing it just means which the plant can use. So when we have this nitrogen applied and it's loose in the soil, meaning that it's not stored in the plant and it's not stored in the microbes, to contaminate local water systems, which is where we hear about high nitrogen in our runoff or in our rivers. And it's also more likely to escape and move back into the atmosphere as gas, which just means it's no longer available in the soil. So then third, when we have this excess soil nitrogen, another factor is that it makes the habitats and the soil conditions unlivable for the microbes. So microbes don't do well in a high nitrogen soil where the nitrogen is loose and not being used. And then finally, once the plant nutrients are all used up or they have moved out of the soil becomes dead. And we often hear this term from dirt to soil. Gabe Brown kind of came up with that and has a book titled that. But really, it's true. The soil becomes dead. We don't have a lot of living organisms in the soil. And the plant then needs nutrients to continue to grow, which, as farmers here know, requires additional fertilizer and additional money in this world. So this is a sort of general overview of how the algae is working. If you are having specific questions about this, please either type them in the chat or write them down, remember them for our Q&A. I'd be more than happy to answer those. But now it is my pleasure to introduce Dr. Jerry Hatfield. I'm going to let him give you a little background on himself. And then we will talk about improving production efficiency. And I encourage all of you, again, to type your questions in the chat for Dr. Hatfield or to write them down. And we will give you all time to have a discussion after he's finished speaking. So over to you.
And thanks, Camille, and thank Yazzie for this opportunity. And I just give you a little bit of background for those who don't know me. I'm a native Kansan. Got my bachelor's degree out of Kansas State University in the field of agronomy. And got a master's out of the University of Kentucky in agronomy and with really especially in crop physiology. And then I switched schools again and got my PhD out of Iowa State in the area of agricultural climatology and meteorology with a minor in statistics. My first career job was I was on the faculty at UC Davis as a biometeorologist because my background is really about looking at this intersection between biological systems and the physical environment or the environment in general. And so I had both a teaching and research career, and I worked up and down the Central Valley and the Napa Valley and the Sonoma Valley in California to look at all of this. And then in 1989, I transferred, or 1983, I left university and went to USDA Agricultural Research Service as a research leader of a plant stress and water conservation unit in Lubbock, Texas. And then in 1989, became the laboratory director of the National Soil Tilth Lab, and we later renamed that to the National Lab for Agriculture and Environment. Over my whole career, which spans a couple decades, more than that, I've really had this interest in done a lot of work in production efficiency, things that Camille talked about there in terms of how the plant's responding, but looking at how efficiently does that plant use water? How efficiently does it capture light to convert it into all the things that grow there? And then extended that into looking at things like nitrogen use efficiency. How much production are we getting per unit of nitrogen that plant uses? And so right now, we're looking and saying, how does water, light, and nutrients all come together and saying, what can we do in terms of overall production efficiency? As a side note, I retired in January of 2020 and have spent the last 3+ years basically working with producers on regenerative ag and analyzing different production systems. And right now I'm working on how different crops fit into the environment and all of this. So I've stayed very active in retirement in terms of the science and how we can do this, because I have a real passion to help agriculture. What that's led us to is really to start thinking about this whole idea of soil health. And, you know, it's become I was the first laboratory director of the National Soil Tilth Lab. And tilth was one of these interesting terms that was out there. Everybody knew what it was, but nobody knew quite how to quantify it. And we replaced tilth with soil quality. We've replaced soil quality with soil health, because we keep searching for descriptors that says, how does the most basic thing that we use in agriculture really begin to change? How does it respond to all the different pieces that are out there? So we look at this indicators of soil health. One of the first ones is microbial activity. And a number of years ago, I built this diagram to explain the changes that go on within this. And if we think about changes in the soil as being a staircase that we go up, the first step on that staircase is biological activity. We have to promote biological activity in that soil to get it to change positively anyway. We can always change it negatively by doing all sorts of different things to it, but a positive change requires biological activity. Biology wants four things. It wants food, water, air, and shelter. The same basic things that you and I want. We want to be fed, we want to have water to drink, we want to have air to breathe, and we want to be sheltered. But in the biological activity, when we think about it in the soil, is that the invisible processes that go on, things like organic matter turnover. How does those microbes change plant material into organic matter, all the different things that are going on there? How do they release nutrients into that soil to make it more available to that plant out there? Those are things that we know are going on. We can detect them, but it's hard to see them. But what we can see is the improved soil structure and improved water availability. And so when we start looking at all of these different pieces, we see indicators of soil health in terms of microbial activity. A lot of the measurements that are made there are really in terms of CO2 that comes out of that soil, because those are aerobic systems. They're just like you and I. They take oxygen in, they use that as part of the metabolic process, and they give CO2 off as part of the respiration. So we see that as an indicator. We see organic matter changes. And one of the things in organic matter changes that Camille mentioned is this change in color of soil. We go from maybe a very light colored soil to very dark colored soil. We see these changes occur quickly across this. The other thing we see is nutrient availability. One of the things that, in terms of looking at nutrient use efficiency, is that because if it becomes a biological system, that those nutrients become available all season long, as opposed to a commercial fertilizer in which, when that supply runs out, we see a deficit. And we see a lot of improvements in plant growth and sustained plant growth late into, say, the grain filling or fruit setting period, because the biology is making those nutrients available. And then finally, the last piece of this that we see is aggregate stability. The more we have the biological activity, the more we are changing that soil and making those stable aggregates that not only help in that water retention, but water storage, but nutrient storage, nutrient cycling, all these different pieces. So when you look at all this in terms of indicators, is that these are things that we know how to measure. We just got to figure out really where they begin to all fit together relative to plant systems. That's why we still look at these indicators on production efficiency. Is that system become more water use efficient? Does it become a more nutrient use efficient? Does it become more light efficient in terms of changing this? So Camille, let's just go to the next one. And you look at this, and what was interesting is that this paper appeared about 4 years ago, but it's by Wishmeyer and his co-workers. They surveyed all the literature around the world and said, what are the real factors that cause soil organic carbon to change in soils? And so they started looking across all the different experiments that have been done, all these different pieces, and saying, what are the most relevant factors that are out there? Well, the first factor, just by the length of the arrow out there, was microorganisms or biological activity, fauna within that soil. And then you get to the next one, which is texture or clay content, because the finer, the more surface area, the more different dynamics that go on with the biology. And then we get into land use and management. How is that land managed? What do we do on it? And then we get into vegetation, then climate, topography, how that soil is situated relative to the Earth's surface. Finally, parent material. You see, if you remember the soil forming factors, that a lot of these are very close to that. And then finally, it was the physics and chemistry. And what's interesting is that for a long time, we kind of reversed this. We thought that if we understood physics and chemistry, we'd know how to change organic matter. And in reality, we need to understand the biology if we're going to change the soil out there. And so this is really reinforces that whole ladder diagram that I gave you before, and saying, you know, across the world, we've now begun to realize the importance of biology and how do we manage that biological system and how does it interact with all the other pieces that are going on. So let's go to the next one, Camille, because now we get into this energy flow path. And if you think about this carbon energy flow path, is that we have an abundant resource. That abundant resource is the sun. And the whole process of photosynthesis, my corn plant went away, Camille. I don't know what happened, but we take sun, and in the process of photosynthesis, we take CO2 and water, and we convert that into a simple sugar, glucose. And that glucose really forms the whole process of energy within this system. Because what we really begun to discover is that agriculture and plant growth is in the process of energy capture. And what we're doing is really taking that sunlight, CO2, water, putting it into that plant. We're moving it down the stem. We take part of it and move it out of the roots, and those become root exudates. Those are the sugars that basically feed the microbes. They feed the soil fauna. Within that whole system, it's really an energy process. We've always talked about how efficient that plant is in terms of capturing light, which is energy. Water evaporation back from that plant into the atmosphere is energy. But now we put it into a context of how much energy is going into that soil. And that energy then can get divided into a couple different things. We can cycle nutrients or we can cycle carbon. And cycling nutrients, we take care of our plant nutrition. And when we take care of our plant nutrition, we also take care of food nutrition. One of the things is, what's the relationship between soil health and human health in terms of this overall process, to say, if we produce a more nutrient-dense food, what does that mean for human health? What does that mean for us in terms of being able to supply the nutrients that we need? And I think this becomes one of our major challenges going forward, is saying is, yeah, it's all well and good to grow these plants, but what are we doing in terms of creating the most nutrient-dense foods that we can have out there? In the carbon cycling, the other side is that we put it into ecosystem services in terms of either water quality, reducing erosion, all these other pieces that go with that. And so we take that CO or that food and we put it back through, we make fuel out of it. We either make fiber out of it, we make feed out of it, or we make food out of it. And so all of these different pieces. And so the chat just showed that nutrient-dense foods are basically more nutrients per calorie. And just think about it very simply. It becomes another measure of efficiency in all of this. And we don't think about it's really how much protein there could be for calorie. It's how much of the minerals are within that food per calorie out there. And so it's a very simple definition when you think about it, is that for every calorie we eat, and we all spend our time thinking about how much calories we eat, how much we're getting in terms of nutrients. You always hear people talk about hollow calories. Well, now we begin to see that there is a relationship with what we do in the soil and these whole carbon dynamic and nutrient density of our food. So he really began to bring this into an overall picture in terms of production efficiency. One of the things we know is that when we begin to improve the soil, we improve the water, is that we do increase water use efficiency. Just think about it simply as saying, how much crop do we produce? for the amount of rainfall that occurs during the growing season if you're in rain-fed areas. If you're in irrigated areas, how much crop do you produce for amount of water that's applied out there? And when we begin to improve the soil, we've seen improvements of water use efficiency by as much as 40 to 50%, is that we're getting 50% more grain for the amount of water that we apply into that crop. And we, at the same time, we've improved our light capture efficiency because we are capturing the same amount of light, but we're converting it into more biomass or more grain or fruit or anything else that we're harvesting out there. And we also see an improvement in nutrient use efficiency because the biological systems are cycling nutrients, is that we are making more plant for the amount of nutrients that are applied out there, recycled out there in this overall process. So what we're beginning to see in all of this is that we change the soils very dramatically, and we change them very quickly. We've spent a lot of time looking at this system because we have the capability of measuring all these different components. Is that a lot of our systems lose more carbon than they actually accrue. But if you reduce the tillage intensity or you add cover crops, is that we reverse that loss into a positive. And as we improve our carbon content, we begin to change this. And so one of the things that we see in our systems that Camille mentioned is that we change our water dynamics. And once you change the water dynamics, you change the carbon dynamics, and then you change the nutrient dynamics. These are all stacked things, and they're occurring at different rates as we change the soil out there. So if we think about what we do in changing water, is that that's an immediate effect. We grow more plant. The more plant we grow, the more carbon we put in. The more carbon we put in, the more nutrients we cycle, because those are all biologically driven. And so we see all of these different pieces. And so we've seen over time that within 2 years of making the change in the soil, we could actually double the microbial biomass in the upper 6 inches of the soil. That's pretty dramatic when you begin to think about the degree in which we can change some things. So we begin to look at all this. We need to understand that this simple little diagram, you can go back to that ladder diagram, Camille. You think about all these different pieces that are going on and changes out there, but that first step is now beginning to understand, how do we treat, how do we understand, and how do we quantify the biological system within that soil. So with that, that gives you kind of an overview of production efficiency. How do we think about this? We can have a great conversation about ways in which we ought to start looking at our soil and what we can do in terms of improving the capacity and the capability of producing the food that we all need and the type of food that we all want to eat. So Camille, I'll turn it back over to you.
Thank you so much. I really appreciate listening to you. I apologize about the corn photo. I'm not sure where that is, but I tried to improvise. So Yazzie and I realized that we never introduced ourselves at the beginning of the webinar. So we're going to do that real quick, and then we would love to take your questions. So Yazzie, why don't you go first?
Hi, everyone. So I'm Yazmeen Rodrigues, and I help run the partner program, which we've created at Enlightened Soil to help communicate with different communities better by finding these local champions. And then I also do marketing for Enlightened Soil. So I run the Instagram, which you should follow.
And I'm Camille. I am the Director of Education currently, and also help with a lot of grower support and farming operations. I am a regenerative livestock farmer myself. I live in Michigan and absolutely love what I do out in the field. And we together work in education and research and sales, and have a wonderful team, some of whom are here tonight. So with that, we would love to take your questions. Feel free to ask Dr. Hatfield anything about his slides or about his life and career. You're also more than welcome to ask us any questions about EnSoil specifically, or the company, or what we offer. It's really an open floor. I invite you to type in the chat or unmute yourself. I will be reading out the questions from the chat if that's more comfortable, or if you'd like to share your voice, we'd love to hear it. So let's see. We have, for Dr. Hatfield, is there a nutrient density standard that you are aware of? Thanks, Darcy, for that question.
Yeah, probably. We have standards for everything, but I think this is, what level should we have for different nutrient density standard for a grain like wheat? It'd be entirely different than a nutrient density standard for a fruit or anything else that's part of this. So I'll have to admit that I haven't spent a lot of time looking at whether they're standards or not, but looking and saying, how does soil impact the nutrient density in there without looking and saying, are we at the have we reached the threshold, or what is our potential on a lot of this? So that's an excellent question, and it really we can begin to look at that and say, what should our optimum be, or what should the maximum be on this?
Great, thank you. From Willie, we have Bionutrient Food Association has done most known work on nutrient density, but also Dr. Fred Provenza and continued research out of his research work. Thanks for that, Willie.
I actually have a question. So you were talking about the amount of energy that the soil can take in from the crop. Mm-hmm. And me and you talked a little bit about this before, but I was wondering, what is the difference between the amount of energy it takes to build the soil versus the amount of energy it takes to deplete it?
Yeah, I mean, we're all in the energy business. I mean, and you think about the energy that it takes to deplete it is one of our biggest culprits in depleting soil or degrading soil is tillage. Because every time we stir that soil up, we basically increase microbial activity. For a short burst, we get these big bursts of CO2. Don Rakowski did a lot of from Minnesota did a lot of measurements with a system where he actually put a chamber over different tillage operations and found out, just as soon as that tillage instrument went through, there was a big burst of CO2, and you could watch that. We've seen that because we don't do little chambers, we do big flux measurements, and we see that same thing. When we till the soil, there's one thing that tillage does that everybody knows, and that's dries the soil out. You see this massive increase in evaporation, but we also see this massive increase in CO2. So this continued use of tillage operation. The other thing is that we can actually have biological systems with reduced tillage that degrade dead soils if they have they're not putting as much carbon in as we're taking off, and so we can actually deplete the soil that way. Across Iowa, we estimate that since the prairie was broken and we began to farm it, we've probably lost 60 to 70, maybe as much as 80% of our original organic matter has been lost until today. And so the degradation process is just been continual over time. The other piece is that now when you think about the flip side, because we did run an experiment because we were seeing all this carbon loss in our conventional systems, we converted a system over in 2016 to a reduced tillage and a cover crop system. And within the first year, we went from a negative carbon balance to a positive carbon balance because we'd reduced the tillage intensity in that. And that's where we begin to see that big increase of microbial activity in the upper 6 inches of the soil. So all of this is we can grade that soil, we can begin to increase it, but we have to realize that our increase is coming because what we're really promoting is a healthy biological system. It is really all about the energy that it takes to sustain that. Just like you and I, if we overeat, what happens? We gain weight because we're taking in more calories than we need for maintenance. The same thing is really happening in our soil, is saying if we don't feed them enough, what happens is that they don't have the energy to do the thing. So we have to have a given set of energy for maintenance. And if we want that soil to improve, we've got to add even more energy, or a biological system that's very capable of extracting that and putting it in there.
Thank you. Great question, Yazzie. So we have a couple of questions in the chat. The first one I can take here about EnSoil. So Darcy asked, in Canada, they're currently using a micronized algae extract, and they're wondering what makes a good algae and why would EnSoil be the algae of choice. And I clarified to ask whether it's living or an extract, because that's an important distinction on the market right now. So EnSoil algae currently, as far as we know, is the only alive cell that is an algae cell that's on the market in the US. And the reason that this is really important is that the reason that EnSoil has this distinction has to do with the mechanism of it being alive. So when you use an algae extract, you're getting a lot of the benefits of the algae properties in stimulating the microbes and helping that plant draw down more carbon. But the difference is that with a live cell, the live cells actually are transporters. They're like taxi cabs or buses or however you get around in your day-to-day life. The algae cells are acting like that in the soil for the microbes. So we have a really fascinating study with Dr. James White at Rutgers University, where he looked at our algae, the EnSoil algae, under a microscope, and he saw that when the algae goes into the soil, it actually engulfs microbes. It fills itself up with soil microbes, and then it moves into the plant root. And for those of you scientists, that is called endophytic. So the algae cell moves into the plant root, and then it lets everybody off. And so what happens is all these microbes are now colonized in the plant root, where they're going to be doing a lot of necessary work for that plant. And this transportation mechanism is one of the key things that keeps us apart from other algae products that are extracts. So I hope that answers your question, Darcy. I'm happy to explain further if you have more clarifying questions. Let's see, Jim, we'd love for you to share your question about water evaporation again. We did not get that. I'll go to Clay. So, and Yazzie just put that Dr. White study in the chat for everyone. So Clay says, how is it kept alive in storage, shipping on the farm? So great question. As Yazzie said in the beginning, Dr. Taylor was able to essentially figure out how to keep the algae shelf stable. So we grow the algae in a lab in bioreactors, which are essentially large glass chambers. We use structured water and oxygen and light. That's it. And the algae grows in there, and we get to a stability of cell count, and then we bottle the algae. The algae is then shipped and stored. Currently, we're recommending that it's stored in the refrigerator. That being said, mainly as long as it's out of direct sunlight and direct heat, the cell count will be stable. Currently, we have a shelf life of about 4 to 6 months for the algae to stay alive. And the reason that it's able to stay alive is that Dr. Taylor was able to figure out how the algae can be both autotrophic and heterotrophic. And what those terms mean is that an autotroph is something that uses sunlight to produce energy. So that's our plant world. And we humans are heterotrophs. We eat food to have energy. And we don't need sunlight. So we do need sunlight, but we don't need sunlight to grow. And so the algae is able to actually do both. And so that's why the algae can stay alive in the bottle, in your garage or in your fridge or wherever you're keeping it. Okay, Jim's question. Is water loss from tillage, I think Dr. Hatfield, this is for you. So is water loss from tillage the same in cool conditions such as fall tillage versus springtime tillage, referring to your work on 1 pass equals 1 inch of precipitation.
Yep. Jim, thanks for that question. And again, good to see you on the webinar. But if you think about the dynamics of what causes evaporation, there's basically 3 main ingredients. One is the amount of energy that's on that soil surface. One is the humidity of the air, and one's wind speed. And those 3 things really determine the rate of evaporation. If we think about a fall condition, is that we're in a point of which we've got much less solar radiation occurring. The sun's at a different angle in there, so we're not getting as much energy on a square foot or a square yard of soil. Plus, we're also cooler. We'll be cooler at night. We tend to be cooling the soil down, and then the wind speeds can be stronger, but it's basically a radiation and a humidity dynamic in the spring versus the fall condition. And so, yeah, if we do tillage in the fall, we're going to see less evaporation. We were always worried much more about why we're doing it in the spring, and we discovered this a little bit by accident, because we have all these measurements or capability of making water vapor measurements in the atmosphere, is that we would see these puffs of increases in atmospheric water content. And, we hadn't rained or anything else, but what we did discover is that producers were tilling their soil. And so that 1 pass, instead of 1 inch, Jim, it was really a 1/2 inch of precipitation. If you put 2 tillage passes, you've already lost 1 inch. But if you think about this, is that where we're losing that inch of water is right in the seed zone. And so what we're drying out the seed zone and making it potentially in certain years much more risky for that plant, because it doesn't have the moisture to germinate that seed, keep it going after it germinates and everything. So yes, there is a difference between fall and spring, and it's really due to the physics of the process that is going on out there.
Thank you. Great question, Jim. Matt is asking, what are your thoughts on Johnson-Su bioreactors and DIY fish hydrolysate? So I can say my opinions, and maybe Dr. Hatfield, you want to share yours. We typically say that nothing in regenerative ag is a silver bullet. So there's not going to be a one-stop shop for a solution to your soil or any of your challenges that you're facing on your farm. I myself as a farmer know that in day-to-day, there's always things that come up. There's always different things that can help. So we are definitely big proponents of any biological in the system, whether it's a fish product or a compost or a fungi or whatever you got. I think that adding biology to the system is important, including the EnSoil algae, and all these biologicals are going to work together and also work in different ways. And so figuring out, depending on what you're growing and where you are and what your soil is like, some things might work better for you than others. And we're definitely big proponents of trial. Try it out, see how it is, make your observations, come back, maybe try it a little bit differently next time. So that's sort of my general opinion. Do you want to share yours, Dr. Hatfield?
Yeah, I mean, it's you look at all this, and Camille did a great job of bringing one important piece into this, is that Johnson-Su, the fish hydrolysate, is really a way of capturing that biology and concentrating it. Think about it that way. When you think about that tea that's coming out of a Johnson-Su bioreactor, is that you're promoting all that biological activity. Remember what Camille said about how they grew their algae in terms of tanks in which you're basically concentrating it. These things are occurring in nature. They just don't happen to be very concentrated, and that's why we don't see an impact of them. So when we think about bioreactors that increase those so we can put it into more concentrated form, we begin to add really very strong impacts on that. And people see positive impacts of this. And again, it's the biological activity that's out there. Because remember what I said about energy, is that if we want to grow or change the system, we've got to supply more energy than is required for maintenance. And these systems are not silver bullets, but they are ways in which we now beginning to understand the system, because let's face it, we haven't been in the soil biology business as long as we've been in the physics and chemistry business in looking at soils. And so we have a lot to understand and a lot of knowledge to gain about how different populations behave. What's this ratio between fungal and bacteria within the soil? All these different pieces that come together just need to be understood. And so I don't think that we have a lock on how all this is responding, but we do know the principles now. We know the principles of being able to supply more energy. How do we get it there? Is a way of delivery. Cover crops, for example, really, and think about from the energy process, all we're doing is energy to put excellent into that, and then any we have that's so we need to keep that piece
Awesome.
fed.
Melissa has a question. Says, regarding surface soil effects of tillage in the low precipitation zone of the Columbia Plateau in the inland Pacific Northwest, tillage has historically been used to create a quote unquote dry mulch to block evaporation from the seed bed. But even under these extreme conditions, producers have begun to adopt no-till systems to increase the soil water holding capacity over time. Yeah, that's great. Do you want to say anything about that?
Oh yeah, that's one of my favorite questions, and it really is about this whole process. And it goes back to a little bit of the question that Jim asked about spring versus fall conditions in terms of tilling that soil and evaporation rate coming out. Because one of the things that we see when we begin to add that residue layer from the no-till on top of that is that, yes, we create a dry mulch. And one of my first classes at K-State in terms of soil management is that we actually had two books on soil management. One was for the eastern part of Kansas, and one was for the western part. And we talked about a dry mulch layer that basically you increase the air in that layer in there, so we reduce the evaporation rate. But at the same time, when we've got that bare soil out there, we're creating an extremely hot environment that basically cooks the biology out of that soil. So when we start going to a no-till system with a cover in there, we do two things. We shade that soil surface. We create a very calm condition or no wind speed near the surface, so that any rain that gets in there, we reduce the evaporation rate, we change the energy balance. We begin to allow that biology to work slowly over time, because again, we're in a very dry environment, but it does work, and we begin to see this. So when doing all these different pieces allows us to come together, and I could give you a whole lecture on the of all of this, because we've spent a lot of time looking at really what are your really indicators of changes out there. And so, and producers really begin to realize that maybe that energy that's spent creating the dry mulch isn't as effective as not putting that diesel energy in and allowing the system to respond differently.
Great. These are awesome. Well, thank you so much for your participation. Darcy asked, have you done research or trials with stackings? Is it beneficial to add biological, humic, etc.? So we currently official data published on this, on like combinations of biologicals. We do have a lot of growers who use combinations of biologicals and incorporate that into their personal data. So yes and no. If you are curious about the details on that, please reach out to me, Darcy. I would love to chat with you specifically. We do have some work going on with some people using more sort of DIY biologicals from their home, and also more what would I call them, commercial compost extracts, teas, etc. We do work with 2 vermicomposters, and they have seen very good results on feeding their worms with the algae. So I'm also happy to share that data with you if that is something that's of interest. Jim says that pass in the spring, isn't that having a profound impact on capillary water through the growing season?
Oh yeah, anytime that we disturb that soil surface, we break up all the capillaries. We break up all the movements in there, but we've dried that upper 2 inches of the soil out. So I mean, it's it really does need to look at it. I mean, we're going to see differences if we got a loamy soil or a sandy soil versus clay soil in all of this, and we'd see different rates and different responses of all of this in terms of the dynamics going on.
Great. We have 2 questions from the survey. I'm going to take the second one first and answer it quickly, and then I'll pass it over to you. So Randy wants to know, is there anything InSoil does not mix with? Currently, we publicize that you cannot mix InSoil with fish-based products like fish emulsions. I myself do that all the time on the farm and have absolutely no problem with it. So I would say proceed with caution. The biggest key that I want to mention is do not leave products in your tank overnight, because you will get congealing and it will be very hard to get them out. So if you're going to apply, say, a fish-based fertilizer and the algae, apply them together, like put them in the tank at the same time, go out and spray, rinse your tank when you're done. Same thing with any other products. I would recommend that just for ease sake. I've cleaned a lot of sprayers and taken a lot of time doing that, and it's not always fun. But yeah, any other products are totally fine to mix with InSoil. And then from Willie, what are your top 3 favorite indicators of soil health and why?
Okay. Yeah, that's a great question. My favorite indicator of soil health, and I tell producers, if you want to know if you have soil health, go out to your field and look in your field after a 2-inch rain. Because if you look at that soil surface and you see little rivulets moving on where it indicates water is moving, is that you do not have infiltration. And because it may move to a different spot in an infiltration, but you should be able to absorb 2 inches in that soil without any soil movement at all. So that's one of my favorite indicators. The other one is basically take your spade or trowel. I think every producer ought to have a spade, dig their soil up and look at, and just this crumbly structure. One of those is aggregate stability. And when you're also digging it up, you see the color. You can see color changes in the soil as with biological activity. I know that in my stay at Lubbock, I was working on some wheat cotton rotations and everything. And after a couple years, and that white caliche type soil in West Texas, we could already begin to see a change in color in the upper ½ inch. Because we were adding more organic matter into it. So I think that those become the things that you can look at. I mean, there are a lot of other measurements. So we can make CO2 burst. We can do all the different things that are out there. But these are things that you can visually see in all of this. I think the more people can visually see the indicators out there, the better they're off than saying, really what's happening.
Great. Willie says, how does Dr. Jerry like approaching an assessment of ecosystem functions, strengths, weaknesses, etc., for ways to best appropriate tools management to meet the context for boosting the soil aggregation process?
Yeah, I mean, that's ecosystem functions. Agriculture has spent all of its time looking at one ecosystem function, and that was provisioning. How much do we produce? But we spend little time thinking about the regulating piece of it. And that is, what are we doing with water quality? What are we doing with air quality? What are we doing with all these other pieces? That's where these efficiency parameters come into it. We also talk about the indicators in terms of supporting function, the pollinator habitat. That's probably the one of the easiest ones to think about there. The other one, in terms of the sociological aspect of ecosystem functions, gets a little harder to quantify because, but it gets back to, and I think that the last question bridges into this when we start thinking about, how does that soil look? How does that plant look out there? I think there are a lot of sociological benefits to a producer to see that very healthy looking crop throughout there, seeing bees and butterflies in that system. So you can begin to have a social value of all of this as well. And so when we start looking, however, at these tools and management, that's why I get back to this thing of production efficiency. You know, have we become more efficient in capturing light to convert it into things that we want, as well as protecting the environment? So this balance between production and environment becomes very important. You know, how do we put it into that? And again, that last part of that question is put it into that aggregation process. It's really that aggregation process is supporting that biological function and keeping it maintained. And I'll go back to those four things that it wants. It wants food. We got to have the energy to make it work. We want to have air. We've got to have sufficient oxygen going into that soil because if we take it aerobic, you know, we've got a problem. They obviously need water. All biological systems need water. But then the fourth piece is this that often gets overlooked is the shelter piece, is that biology is just like you and I. We don't want to be disturbed. We don't want change any more than anybody else. And so the more stable we can make that environment, the better that all the other three things work. But we often put shelter clear at the end, but shelter needs to come and basically saying, let's create a very stable home for all this to happen in.
Great, thank you. I just want to say we are at 7:58, so please hop off whenever you need to. There is a link in the chat for the big soil health event, which Dr. Hatfield will be speaking at, a lot of other legends, as Yazzie says. You can use the discount code EnSoil to get percentage off of your ticket. So please go to that link and register if you haven't already. I'm happy to stay around and answer the rest of the questions in the chat for those of you that want to stay past 8 o'clock. But again, we appreciate you all coming and we hope to see you at our next webinar. Stay tuned and for the follow-up email tomorrow. So Clay, how is your algae applied, at what rate, when, incorporated in soil, what time is best to apply, at what cost? Great questions. So the algae is applied in any way that you can get water to your plants. You have a sprayer, pivot, drip irrigation, flood irrigation, a watering can in your house, whatever you got, backpack sprayer, anyway, it really doesn't matter. The rate is 8 ounces of the algae per acre of land, or 1 teaspoon of the algae per gallon of water. So depending on how you're applying, you're going to use a different measurement based on your land or your water. We recommend that you apply at seed if you are seeding. That's when you're going to get the most benefit to the seed to sort of set that seed up for success. If you have perennial plants growing, we say typically 3 times a season is a good measurement. We have people who spray more than that. We have people who spray less than that and have good results. So it's really dependent. It does get incorporated into the soil, but it's a foliar spray. So it will land on the leaves and it will also land on the soil. The algae don't move far. They really stay where they're sprayed. And typically they're doing their work in about 1 to 3 inches deep below the surface level. It is best to apply not when it's super, super hot. So if you live in a place where it gets super, super hot, apply in the morning or in the evening. If not, it doesn't particularly matter. It is important to have some moisture in the ground when you are applying. So if you live in a place that's super dry, try to plan your application around a rain event if you can. We know that that is not always something that we can control. It can be really frustrating, been there, but that's a recommendation. And then the cost, we have a pricing matrix that is dependent upon the amount of algae that you are purchasing. So that can be as low as $9 an acre or as high as about $20 an acre for really small quantities. Darcy, I was reading blogs on your site and one talked about reducing nitrogen and seeing results. What is your thoughts on nutrient balancing along with EnSoil? So I'm interpreting this. I think I know which blog you're talking about, and we have seen a lot of results where we've helped folks transition away from synthetic inputs such as nitrogen. And what that typically looks like is, we'll start somebody off using 100% of their inputs and no algae, and then we'll titrate them. So maybe that looks like 80-20, then 50-50, 20-80, et cetera, all the way down. We really want to meet you where you're at. So this is not a pressurized system by any means. We have growers that use the same amount of their inputs, and they've been working with us for multiple years, and they apply the algae and they keep their fertilizer exactly the same. And that is totally okay. It's really whatever you're comfortable with, and we obviously want you to maintain your yield as best you can. So I hope that answers your question. If you want to clarify that, please ask something else. Question from Wendell. I have seen studies where soil amendments are not consistent in their results. Is algae different? If so, what studies have been done? This is a great question. I think any scientist and any researcher has seen data that is not consistent in results. That's kind of the nature of doing research and having data and results. So I would say that we're definitely not an outlier to that. We are still learning a lot about our product, and we rely on you all as growers and as observers to help us learn about our product. We do have consistent data in a lot of areas, and the 4 mechanisms that I explained at the beginning of the webinar are definitely things that we can back up with consistent data. Is that consistently consistent across every single time we've used the algae? Of course not, because climate is changing, weather is changing, life is changing, but we do feel that we can back that up with peer-reviewed evidence.
Camille, that one question on there that's about the adjustments on soil texture. Remember, back on our drivers of organic matter change is that texture of the soil was the second factor in all of this. And so, yes, we will see textural differences because of surface area. You think about a sand particle out there, there's little space for that biology to hold on to. But you get a clay particle that's a lot of surface area, a lot of spaces for that biology to hide and glom onto, really will have a different impact. And so we will see a textural response. We see that across fields. When we look at some of the responses from cover crops or anything else, is we will see that impact. So texture has to be looked at, and we'll see consistent or inconsistent results across the textures just because of the dynamics of soil that's out there.
Thank you. Great. We appreciate all the gratitude. We're so glad that you're all here as well. I think the last question we have is from Jim. Can you comment on the phenomenon of microerosion and the impact on the soil ecosystem?
Yeah, the micro erosion, again, you're looking at that soil moving in apart. What's the piece of soil that moves? It's the really small particles that move when we talk about sediment, and that those we actually have a diagram where we talk about stable versus unstable aggregates. Raindrop hits an unstable aggregate, which breaks into sand, silt, and clay. The silt is basically clogging up all the pores, and so that's why we reduce the infiltration rate. We keep that stable aggregate, keep those pores open. So yeah, the micro erosion is basically taking that silt and moving it in and clogging up those capillaries so that we prevent erosion. But we also prevent air exchange. And we don't often talk about the air exchange in soil, but that's as critical as the infiltration of water, as the movement of gases out there. And so those have to be looked at, because all of those pieces that are out there. So my mantra in all of this is that moving soil becomes non-productive soil, and it's just a matter of scale of which it's moving.
Great. And I think that's a great note to end on. So we wish you all a wonderful evening. And again, thank you so much for joining us. Thank you, Dr. Jerry Hatfield, for being our guest. We really appreciate you and your time. You all to attend the Big Soil Health event. I will be there. I would love to meet you. and chat about soil and nerd out about farming stuff or whatever you want to talk about. So definitely come and introduce yourselves if you are attending. We will have another webinar in 2 weeks, and we hope to see you there. Again, look out for that follow-up email tomorrow from Yazzie. You want to say anything else before we sign off?
Just want to say thank you, Camille, for the answering the questions in speedy time, and Dr. Jerry Hatfield, thank you so much for sharing your brilliance with us. And yeah, everyone, look out for my follow-up, and you'll get the recording and all other sorts of stuff.
We appreciate you all. Please go spread this wonderful knowledge about regeneration to your communities. That's how we make change. Have a great night, y'all.
Thank you.